All right, hello everybody. Welcome to your second genetics video. Human chromosomes. Human chromosomes are made up of DNA, and DNA forms this double helix that we see in the image here. That double helix and its structure is going to be covered in more detail in our third genetics video, but we're all familiar with that twisting ladder form and the DNA code is made up of A's, T's, G's, and C's. Well, in order to form a chromosome, DNA has to coil up. So we have 46 pieces of DNA in every cell of our body. That genetic material is going to coil up and form these dense coiled structures called chromosomes. In order to coil up and not get caught or tangled, the DNA is going to wrap itself around these purple spheres. These purple spheres here are called histones. They're made out of protein. And in groups of 8 called octamers, they'll form these double loops. And these double loops enable the DNA to coil and not get tangled. So the DNA coils around the octamers, forming a nucleosome. The nucleosomes start to accumulate and they will coil up, forming coils. But it's not the end of it there. The coils themselves will also coil up, forming what are referred to as supercoils. Those supercoils are going to be uh, wrapped back and forth to form a tight pile that forms one part of this chromosome. All right. And then you're going to have the identical copy of that DNA that's going to be connected to it at the centromere. So you've got two copies of the same DNA. Right? You have an original and its copy coiled and connected right at the centromere. Right? There are 22 autosomes, and the last pair are known as your sex chromosomes because they're integral in the development of sexual characteristics. We can look at a picture of karyotypes, of chromosomes, using a technique called karyotyping. What we do is we find a cell sample undergoing mitosis and we stain it. The stain will attract and bind to the A's and T's, that's adenine and thymine, of the DNA strand. And so areas that are heavy in A's and T's get really dark, and areas that are lighter in A's and T's and have more C and G, which is guanine and cytosine, well, those are going to be lighter. And a lab technician or geneticist who takes this picture would be responsible for sorting out, either using a digital picture on a computer or old school method. They would take a photo and they would cut it apart and they would kind of organize them that way. And they'll sort them by size and with their homologous pair, looking at the banding patterns and the locations of the centromeres. So they're going to be trying to match each one of these up with their homologous pair. From time to time, they'll notice that there might be an extra or even potentially a missing chromosome. These conditions are a result of a process called non-disjunction. And you're familiar with disjunction, you just haven't heard it called that. During mitosis, when all the chromosomes line up along the midline in metaphase, and we could be picturing meiosis or mitosis, it does not matter. They both line up at the middle during metaphase, and then the copies have to move towards the poles. If for any reason those individual copies don't pull apart and they stay stuck together, you end up with extra DNA on one side and missing DNA on the other. And the result is cells that have one or more extra copies of a chromosome. If that cell that's made is a gamete, meaning a sperm or an egg or pollen or ova if you're a plant, if those are fertilized, they end up with additional or missing copies of chromosomes. And there are a lot of genetic syndromes and disorders that are recognized as a result of having extra or missing chromosomes. So down here, this last one in the red box, this is the sex chromosome. Now, these two look pretty similar. This is a female's sex chromosome because the sex chromosomes in females are XX, but a Y chromosome is really tiny and kind of dumpy looking, like this little tiny one here, right? But we know that we can use a Punnett square to show the probability of any mating event to produce male or female. It's going to be 50-50 because females are XX and males are XY. So if your mom has two X's, and this is going to be important later when we start talking about sex-linked traits, but mom can only contribute X's. Dad can contribute an X or a Y. So technically, your father is responsible for gender determination because only the male has two different sex chromosomes. Now, it's interesting to note that in birds, butterflies, and some reptiles, it's not the male who has two different sex chromosomes. It's actually the female that has two different sex chromosomes. So in birds, butterflies, and some reptiles, instead of using an XXXY system to track it, the females are ZW and males are ZZ. And they only change the letters so that they can make a distinction between the way humans and other mammals do this. But it's interesting in birds, all right, the female determines the gender of the offspring, not the male. But in humans, this is the case. That is not to say that the XXXY system, or with birds, the ZWZZ system, are the only ways to determine gender. And in fact, we know that there are several environmental factors that can influence the gender determination in wildlife. For instance, you're probably familiar with or have heard about how temperature in reptiles can influence gender determination. So the temperature that the eggs develop at can influence gender. Or in some aquatic organisms like the limpet, they have alternating gender lifespans where they actually grow on top of each other and they'll switch gender just so they can make offspring. And you may have heard about clownfish have the ability to change gender. These phenomenon are not uncommon in nature. So it's important to understand that environment can also play an impact or has an impact on the gender determination of an offspring. This diagram here is showing a pedigree chart. Pedigree charts are used to show family relationships and allow us to track one particular trait that exists within a family through multiple generations. The generations are denoted over here on the left There's a P generation, which is the parent generation, shown as the first individuals that crossed. The F1 generation are the family members that are created from that first cross. And then the F2 generation are the offspring of that generation. So we've got 3 generations of family members shown here. We see circles representing females, and we have squares that represent males. Any mating pair will have a line connecting between the two of them, and their offspring will be connected to a vertical line, and they will be sorted chronologically, showing the oldest to the left and the youngest towards the right. Individuals who are half shaded are carriers for the particular trait that you might be tracking. Individuals that are fully shaded indicate a person who is expressing a particular trait, which means that is their phenotype. They physically show the characteristic you're tracking. And then those individuals who have no shading indicate that they're not expressing the trait, and they're also not carriers of that trait. And so we can see and track through a family different characteristics, like, for instance, color blindness. We can make a pedigree chart to show color blindness, which is actually a sex-linked trait, which will be coming up shortly. But we can also track more serious conditions like Huntington's disease. Huntington's disease is an example of a dominant genetic mutation that causes early death in humans. So there's a broad spectrum of characteristics you can track using a pedigree. That brings us to sex-linked genes, and sex-linked genes are the group of genes that are all located on the 23rd chromosomes. The majority of these are X-linked. There are a few Y-linked, but these 3 here are all X-linked. Color blindness, hemophilia, and muscular dystrophy, all located on the X chromosome. If you are a guy and you are XY chromosomes, then you only have one X chromosome. In that case, your X came from your mother. So we're not being gender biased here, saying that these are all from mom. But if you're a guy and you have any one of these conditions, then it's a result of your mom's X chromosome. The Y came from your dad because he's the only one who could offer that to you as a guy. If you're a female, you have 2 X chromosomes. And so as a result, you may potentially have a backup X chromosome that's normal. And that brings me to this color blindness through sex linkage Punnett square that we have here. We can see over here in the key, there's a male who's colorblind, and that person is shaded in, and normal vision is white. A female is going to be shaded, and normal vision is white. All right, here's the father. The father in this particular case is carrying the large C, which in this case is normal color vision. And so this father has normal color vision. This mother also has normal color vision, but she's a carrier for this mutant colorblind gene that she's carrying here. So we'll see, just like in the pedigree, she's half shaded. So you have the colorblind X chromosome that she has the potential to share, shown right here with this little c there, that lowercase c, and then she also shared it here to this potential son. This particular X chromosome with the normal color vision gene can also be shared, but you'll notice that these offspring have normal vision. This particular female has the gene for normal color vision and the gene for color blindness, but she has normal colored vision, and she may not even know that she's carrying that gene for color blindness. And she wouldn't know until the next generation, typically, if she were to have children with a colorblind father, that is the only way they could have a colorblind daughter. But this son, he got stuck because he's the only individual who received the color blindness X chromosome mutation, and he only has one X chromosome. So that's how color blindness through sex linkage works, and it works the exact same way with hemophilia and with muscular dystrophy. This brings me to a technique used for detecting genetic defects in fetuses. A process called amniocentesis can be done that enables a geneticist to examine the amniotic fluid during a pregnancy to look for chromosomal abnormalities. In this situation, under the guidance of ultrasound, so they'll be viewing this at the time of the procedure, a needle will be inserted through the abdominal wall, through the uterine wall, and into the amniotic fluid that surrounds the baby. A small amount of that amniotic fluid will be drawn out and it'll be centrifuged. And so it gets spun around. That's what centrifugation is. And it separates it by density. The fetal cells that are floating around in the amniotic fluid settle to the bottom, and they're able to culture them, and they're all going through mitosis. They're growing and developing quickly, and so they can take a picture of the chromosomes as they're going through mitosis, and they can develop a karyotype. And the karyotype is what we described earlier. It's a picture of those chromosomes sorted out by size and based on their banding patterns. A linked gene. This is the SRY gene, or sex determining region Y, SRY. This region is responsible for coding the production of testosterone receptors on cells, and it's critical during the fetal development or embryonic development, even stages of development in utero. Mutations that result in no receptors, even if you're an XY individual, it will result in female phenotype development. So you can be a fetus whose cells do not produce functional sex determining region Y protein, but you won't develop testes. You'll develop a uterus and fallopian tubes despite the fact that you are an XY person. This condition is referred to as Swyer syndrome, and it happens the other way as well. You can be XX, meaning you're female, and you can develop characteristic male structures in development if the SRY gene has been crossed over onto one of your X chromosomes on accident. So having this SRY gene seems to be a determining factor in the development of male characteristics. This is the distinctive male growth patterns of body hair, muscle development, testes, in the lack of SRY results in the development of ovaries and uterus and fallopian tubes. So this is a Y-linked characteristic. This is gel electrophoresis. It is another genetic technique used to do some analysis of genetic material. In this particular case, DNA is sampled and placed into an agarose gel, which is kind of like a colorless, tasteless gelatin or jello. And after an electric charge is passed through it, the DNA will migrate towards the opposite side. Now, I have a little bit more to show you about this, but the process is pretty straightforward. The genetic material that is sampled has to be treated with a restriction enzyme, which is a special enzyme to cut the DNA at certain sequences. That's going to take a really huge DNA molecule and make it seemingly random sized pieces. So anywhere a CCGG DNA combination might be found, the enzyme will cut it. Well, you're going to have some large pieces and you're going to have some small pieces. The banding patterns that form are a result of different sizes. So the largest bands are found near the top and the smallest bands are found near the bottom. And every individual has their own unique banding pattern. So we can use this as what is known as a DNA fingerprint. We can use this to identify individuals in a whole host of different scenarios. We can use it in criminal cases. We can do it in paternity cases. We can use it for identifying the likelihood of certain conditions developing. So here's gel electrophoresis in a little more depth. This is a step-by-step process here. The DNA samples are copied using a system called PCR. And that just makes lots and lots of copies of a DNA sample. You prepare your gel agarose, and the DNA samples are treated with the restriction enzymes. This cuts it up into little pieces under number 3 here. And the DNA sequence does vary. There are lots of different enzymes that we can use, but you should know that restriction enzymes cut the DNA at a specific sequence. So those cut DNA pieces are carefully transferred to the wells, which are the little holes in the gel. And the gel rests in a buffer solution that has it has some chemicals in it that make it conduct electricity. It's pH controlled and such. So when you turn on the power supply, this negative pole here, the electricity travels from there to the positive pole. And the electric current causes those fragments of DNA to migrate. And they use a what's called nitrocellulose sheet. That is one way that they do it, is they'll take this nitrocellulose sheet, they'll put it over the top of the pieces, and that will absorb the bands in the DNA fragments. Now, once you absorb the bands of the DNA, you have to stain them with a fluorescent probe. And so we've got a couple of things that are happening here. The left-hand side is always used as a marker, or a standard is what they call it. So they'll have known sized pieces of DNA in that sample. And then that way, when a band shows up in the same spot, they know how big that band is. So this band, this pink dash here, would be sizes of DNA fragments that are the same as here. And this band would be the same as this fragment. Now, if the bands, you have the standard row, right? And then you have your comparison row. Like if this is a criminal investigation, you might have the blood from a crime scene in this first row after the standard. And then following that, you might have your potential suspects who you've collected a DNA sample from. And you would run those next to the blood from the crime scene. And if you can find a match, then you can prove with virtual flawless consistency that that individual was present at that scene and their genetic material was left behind. Now, there are ways that this process can be flawed, and it is certainly not perfect, but it is very, very reliable. Here's another chemical process, another analysis technique we use called chromatography. There's different types of chromatography. We use paper chromatography pretty exclusively in our biology classes, but chemistry labs will also do gas chromatography, which is a little more detailed and it's a little more complicated, a process. You have to do it with a machine. But chromatography is a process for separating parts of a mixture. So this is loosely based on how well a chemical dissolves in a solvent. A mixture sample is placed, a sample like this ink spot here in the image, all right? This sample is placed on a sheet of chromatography filter paper, and the paper is just barely dipped into a solvent. Now, solvents can be varied. You can have water as a solvent, you can have alcohol as a solvent. We use acetone as solvents. Uh, these are all liquids that can be absorbed by this filter paper, and it'll be wicked or sucked up through the paper almost like a paper towel that's just barely touching a liquid. It'll draw the water up through capillary action. It's the attraction of the water molecules or solvent. So as that liquid gets sucked up into the paper, it's going to pass right by your ink spot or your mixture. The solvent is going to dissolve parts of that mixture differently than other parts. So some will dissolve really well and it'll be carried right with what's called the solvent front. And that solvent front will be with the top edge of where the liquid has been absorbed up through. The more easily it dissolves, the faster it stays, or the better it will stay with that solvent front. So different chemicals will dissolve less and they get stuck behind. And so over time, it will gradually be separated from the less soluble parts of the mixture. That's paper chromatography or chromatography in general.